Welcome to this explainer. Okay, so you probably already know a bit about DNA, right? But here's the thing, it doesn't just float freely in your cells. It is packaged with absolutely incredible precision. So today, we're diving right in to decode that ultimate biological package, the chromosome.
To truly appreciate what a chromosome even is, we have to start with a seriously massive mechanical dilemma. Think about this for a second. How do you fit a 1-meter-long thread of DNA into a microscopic box, the nucleus of a single cell? I mean, if someone handed you a huge spool of loose thread, you wouldn't just shove it into a tiny container, would you? No way! You'd coil it up neatly. And, well, it turns out your cells take the exact same approach.
Here is our roadmap for today. First, the packaging problem. 2, chromosome anatomy. 3, the genetic breakdown. 4, sorting by shape. And finally, number 5, autosomes and sex chromosomes.
All right, let's hit section 1, the packaging problem, moving from noodles to yarn. Okay, so for most of a cell's life, your DNA is basically just hanging out as these loose, fragile noodles. But on moving day, when a cell actually needs to divide, it needs durability. So let's go step by step. If we zoom literally all the way in, we start with that classic DNA double helix. Then in step 2, to save space and add some real structural integrity, this long thread of DNA wraps around these tiny protein orbs called histones. This complex forms structures called nucleosomes, kind of like tiny beads on a string. Moving to step 3, this fiber undergoes some intense supercoiling. And finally in step 4, it condenses into that classic durable yarn-like chromosome shape you've probably seen before. This super tight packaging is absolutely crucial because it ensures your genetic code doesn't get damaged when the cell splits in two. Moving right along to section 2, chromosome anatomy. Let's map out this structure.
So, when a cell is gearing up to divide, it duplicates its DNA, right? This creates 2 totally identical sister chromatids. And the knot holding them together right in the middle? That is the centromere. But the centromere isn't just some passive physical connector. Its main job is to provide an attachment point for spindle fibers. Those fibers are eventually going to pull the chromatids apart during cell division, making sure every single new cell gets a complete set of instructions.
Now, branching off from that central knot, we've got the arms of the chromosome. But here's the catch. They aren't perfectly symmetrical. The naming convention here is beautifully simple, though, and it's based entirely on size, not position. The smaller arm is always called the P arm, and the larger arm is always known as the Q arm.
And look what happens at the very tips of those arms. These are the telomeres. Honestly, just think of them like the little protective plastic caps on the ends of your shoelaces. In humans, telomeres are actually made of the sequence TTAGGG, repeated hundreds of times over. But why? Well, every time your DNA replicates, the copying enzymes can't quite reach the very last couple of bases. So this repeating telomere padding essentially sacrifices itself. It erodes just a little bit with each division so your actual vital genetic code doesn't get chopped off. If that padding wears out completely, it leads to replicative senescence, which is really bad news for the cell.
Let's look at section 3, the genetic breakdown. What is actually inside?
You know, you'd probably assume a chromosome is just wall-to-wall genes coding for all the proteins that build your tissues and organs. But get this, coding genes make up only about 1.5% of your entire genome. Isn't that wild? The massive remaining majority, about 98.5% of it, is non-coding DNA.
But hold on, do not mistake non-coding for non-functioning. This vast stretch of DNA between the genes is incredibly important. It transcribes other forms of RNA, serves as the origin points for DNA replication, and plays a massive role in regulating gene expression. Basically, it's what turns specific genes on or off depending on whether you're trying to build an eyeball cell or a liver cell. Plus, non-coding DNA makes up those structural centromeres and telomeres we were just talking about. It even includes these mind-blowing sequences called transposons, which can literally physically move around and change their position within your genome. Talk about a game changer.
Next up, section 4, sorting by shape. It's all about the position of the centromere.
We actually categorize these structures into 4 distinct types, based entirely on where that centromere sits. First up, metacentric means the centromere is placed almost exactly in the middle, making the P and Q arms roughly equal in length. Next, submetacentric means it's shifted slightly away from the center, so the arms are definitely unequal. Then we have acrocentric, from the Greek word acro meaning peak, which means the centromere is pushed almost all the way to the terminal region. And finally, telocentric, where the centromere is placed at the absolute extreme end, or the telomeric region.
But here is a fascinating biological quirk for you. Out of those 4 shapes, the human karyotype completely lacks telecentric chromosomes. You literally won't find a single one in your entire body. And just as a quick reminder, a karyotype is simply the visual representation of all the chromosomes found inside a typical cell.
Which brings us to Section 5, autosomes and sex chromosomes, building the karyotype.
So how does this whole shebang come together? Well, in a typical human diploid cell, which is practically every cell in your body minus the reproductive ones, there are a total of 46 chromosomes. But they don't just exist in some random, chaotic jumble. They're neatly organized into 2 sets of 23 chromosomes. You get one full set from your mother and the other full set from your father.
Now, if we line up those 23 pairs, we see a really clear functional division. Pairs 1 through 22 are called autosomes. These have absolutely nothing to do with determining biological sex. They simply contain the genes that code for the proteins making up your physical body. But that 23rd pair, that one stands apart. These are the sex chromosomes, or allosomes, and they carry the specific instructions that determine biological sex. Here's how it works. During fertilization, an egg cell always provides an X chromosome. But a sperm cell, it can carry either an X or a Y. If the resulting zygote inherits 2 full-sized identical X chromosomes, an XX pair, the individual will become a female. If the zygote gets 1 X and 1 Y chromosome, an XY pair, the individual becomes male. So at the most fundamental level, biological sex determination is essentially a flip of a microscopic coin.
Now, if you look at the male XY pair, it's immediately obvious that the Y chromosome is significantly smaller than the X. Because of this drastic size difference, the two are only partially homologous. This means the Y chromosome is literally missing dozens of genes that are present on the X chromosome. As a result, males only possess 1 single allele for certain genetic traits, whereas females have 2.
And this size difference is exactly why certain traits exist the way they do. Genes that are present only on the X chromosome are called X-linked genes. So if a male inherits a recessive allele for an X-linked gene, he will express that recessive phenotype because he has no second dominant allele to override it. This is exactly why disorders like colorblindness and hemophilia are way more common in males. On the flip side, females have 2 X chromosomes, but typically one is largely deactivated at random in every cell. This random X inactivation creates some amazing biological patterns. It's actually the reason behind the 2 distinct colors of fur you see on female calico cats. Different patches of skin have different X chromosomes actively expressing color alleles. How cool is that?
From the tight supercoiling of DNA around a histone, to those protective telomere caps, to the random dance of X and Y chromosomes, your genome is a literal masterpiece of biological engineering. But remember, alongside all that ancient code, you carry entirely unique mutations, sequences of DNA that have literally never existed in the cosmos until you. So, what unique story are your chromosomes writing? Keep exploring, and we'll see you in the next explainer.